I'm Rachelle Stark, and I'm a metabolic biology PhD candidate in the Department of Nutritional Sciences and Toxicology at UC Berkeley. And I research in the NARS lab the role of the liver in Alzheimer's disease. And so, first of all, what is Alzheimer's disease? Alzheimer's disease is a severe neurodegenerative disease that can cause symptoms like memory loss, confusion, misplacing items, swings, etc. And so it's thought that Alzheimer's disease is in part caused by the accumulation of amyloid beta plaques in the brain, specifically between neurons. And these plaques will accumulate and unfortunately cause neuronal death in the brain. And this leads to the shrinkage of parts of the brain that are really important for memory and learning, which is why we see the symptoms that we see. And so there are a lot of things that promote Alzheimer's disease that you might not think promote Alzheimer's disease. And these things include like obesity, high triglycerides, high blood cholesterol, diabetes, etc. A lot of peripheral diseases that are associated with this brain disease. And so you might still be wondering, how exactly is the liver involved in this? Well, the liver is involved in a lot of those diseases that I just mentioned. And there's been a lot of research that's come out that has started to suggest the liver has a bigger role in this disease than we would have thought. And the main paper that became the basis of my project is the synthesis of human amyloid restricted to the liver results in Alzheimer's disease-like neurodegenerative phenotype. And so in this paper, they created a genetic mouse model that expressed amyloid beta only from the liver, so not in the brain. And they wanted to see if this amyloid beta could make it to the brain from the liver. And they were indeed able to see that. So they saw with MRI and with ELISAs that they did detect amyloid beta present in the brain after only being able to come from the liver. And they also saw other Alzheimer's disease-like phenotypes. And so for my project, I decided to look into this even more and try to recapitulate this experiment. So I created multiple mouse groups where I injected these mice with a virus that allowed the overexpression of amyloid beta from just the liver. So instead of a normal expression like the paper did, I'm overexpressing and I'm using a virus to do this, so it should be quicker. And I decided to do different groups of mice with this virus. And so one group I have on standard chow diet, just normal young mice. Another group I have is mice that are on a Western diet. And then I have a third group that is on a Western diet and are also aged mice. So 14-month-old, which is much older for mouse age. And so I plan to look at the livers, the brains, and the blood in all of these different mouse groups and detect how amyloid beta is interacting with other proteins in the blood and how the liver is involved with these Alzheimer's disease-like phenotypes. And so I'd like to just give a huge thank you to my entire lab for supporting me with this project, but also to the ARCS Fellowship for really supporting a lot of my PhD with this and allowing me to have a lot more flexibility in my project. Thank you.